this pathway which is there in front of you, the glycolysis. So I'll be talking about important aspect of glycolysis in nutshell, which will give you a very crisp outlook to this pathway, right? So good afternoon, welcome everybody, welcome everybody. So what about glycolysis? Glycolysis, you know, is meant for ATP formation, ATP production. Whether it is aerobic or anaerobic, you have glycolysis going on. You have glycolysis going on. There are multiple steps. You see. And the workbook is having all these steps, you know, where formation of lactate is ultimately seen. Lactate is being formed in anaerobic glycolysis. Anaerobic glycolysis, right? So, and pyruvate is the end product of glycolysis in aerobic condition, right? So now you must know what all key enzymes are there. There are multiple steps you can see here, and there is a breakdown of one intermediate which is known as fructose 1,6-bisphosphate. This breakdown is by aldolase A or aldolase B, right? Even aldolase C is a very, very important and minor aldolase isoenzyme which is there. Right, so that is going to break the 6-carbon fructose 6-phosphate to DHAP and glyceraldehyde 3-phosphate. So pay attention to this step. And beyond this, only glyceraldehyde 3-phosphate is further getting metabolized, where number of steps are there. You have the formation of ATP at phosphoglycerate kinase step. You have formation of ATP at pyruvase kinase step. And we talk about 2-ATP production at phosphoglycerate kinase and 2-ATP production at pyruvate kinase step. Do you remember all these? Yes, all of you. Go back and join again. Right. So this is called substrate level phosphorylation, bachcho. Substrate level phosphorylation, where direct ATP is being produced. So this may be a question to you. How many substrate level phosphorylation in the glycolysis? You have to say 2. So that is phosphoglycerate kinase step and pyruvate kinase step, and 2 ATP is produced at each of these sites, producing your total 4 ATP by substrate level phosphorylation. And the point where oxidative phosphorylation may produce ATP is dehydrogenase step. This is the point where NADH is going to generate you ATP in aerobic condition. So this is a place of oxidative phosphorylation. But this will be happening only in aerobic condition. In anaerobic condition, this NADH is not going to produce any ATP. Samreen, my attempt is to give you the important aspect of these pathways, right? So that is why I am telling you to mark it. So I am writing it for you. You also write and mark it. Right. So this is about the pathway of glycolysis. Now you must know certain thing like the key enzyme for glycolysis. What is the irreversible step? They are called key enzyme. Key means which decides the direction, right? So the key enzyme of glycolysis is glucokinase, phosphofructokinase 1 and pyruvate kinase. Just make a I will come to that total ATP and all. You just write down the 3 irreversible steps in the glycolysis is going to be glucokinase step, PFK1 step and pyruvate kinase step. Right? And when you're talking about ATP production, that's again a very, very important point. ATP in glycolysis. You must know this table. That when we are talking about total ATP production, in aerobic condition it is 9 and in anaerobic condition the total ATP is 4. Right? ATP is 4. Alright, what happened Kaniksha? Okay, Venkatesh. Alright, yeah, I will not pay much of the attention to the chat box. Try to focus on the class, right? You already have the workbook with you. Just jot down certain important points which I am just displaying you here. Right, so total ATP in aerobic condition is going to be 9 and the split goes like 4 plus 5. 4 plus 5. 4 is the substrate level phosphorylation and 5 is the oxidative phosphorylation, right? And when we talk about 4 in anaerobic condition, it is going to be substrate level phosphorylation. But when we are talking about net ATP, you have to reduce 2 ATP from these numbers. Why? Because 2 ATP is utilized at the initial stage of glycolysis at this place and at this place, right? So you must know where the ATP is utilized. It is glucokinase or hexokinase, and number 2, it is phosphofructokinase 1 step. So 1 ATP is utilized at these steps. So whenever we are calculating the ATP number, from total number we have to deduct 2 ATP, whether it is aerobic or anaerobic. So total ATP in aerobic is 9 and net is 7, anaerobic it is 4 and net is 2. All right, very good. So these are numbers of ATP. Now the key point in glycolysis, if anybody asks you, though nobody is asking you this now, but it is important for you to know that PFK1 is acting both as a rate limiting enzyme and a committed enzyme. Rate limiting enzyme which determine the rate of the biochemical pathway. So it is PFK1 which is acting as a rate limiting enzyme. And when we say committed enzyme, that is also incidentally it is PFK1. So committed enzyme, when we are talking, it should be irreversible step. Number 1 should be irreversible step, and number 2, it should produce something, some metabolite which is committed to that pathway. So if you talk about glucokinase, you may ask me, oh, glucokinase is also irreversible, that will be committed. But no, glucokinase is converting glucose to glucose 6-phosphate. And please understand, glucose 6-phosphate is not committed to the glycolysis. It can be utilized for glycogen formation. It may get utilized in glycolytic pathway. It may get utilized in HMP shunt pathway. It can get utilized into the uronic acid pathway. So the glucokinase, though it is irreversible, it cannot be a committed step of glycolysis. But when we talk about PFK1, PFK1 is converting what? Converting fructose 6-phosphate to fructose 1,6-bisphosphate, right? You must remember this. You see the glycolysis, this fructose 1,6-bisphosphate is getting utilized only in the glycolysis. There is no taker of this fructose 1,6-bisphosphate, and hence PFK1 is fulfilling both the aspect of being the committed step, number 1, the irreversible, and number 2, it is going to produce your fructose 1,6-bisphosphate, which is utilized only in the glycolysis. All right, very good. Very good. So you have to write all these things in workbook itself. Mark the pathway in this fashion in the workbook itself. That's a point. That's the reason why the workbook is provided to you, right? Okay, now coming to the next important point, which is very, very important for you to note, that whenever we are talking about PFK1, it's an allosteric enzyme having positive and negative allosteric factors. So you must know what is the positive factor for PFK1 and what is the negative factor. Every time I ask you to remember this is a very, very important point. And the positive factor for PFK1 is going to be fructose 2,6-bisphosphate and AMP, and negative factor is going to be ATP, citrate and proton, right? So it is very logical to understand when AMP is there, that is energy deficiency is there, it will try to stimulate the glycolysis, upregulate the glycolysis, right? So it's acting as a positive allosteric factor for PFK1, and negative factor is going to be ATP, because already if the cell is having a lot of ATP, there is no need of glycolysis, right? So that time the ATP will act as a negative modulator for PFK1, negative allosteric factor. So this you remember, and this fructose 2,6-bisphosphate is something very important, which is a positive regulator for PFK1. And I add here, you write down here somewhere. Okay, when we are talking about PFK1, the fructose 2,6-bisphosphate, as you just have seen, is acting as a positive factor, positive allosteric regulator for rate limiting enzyme of glycolysis. When you're talking about gluconeogenesis, you have counter enzyme fructose 1,6-bisphosphatase. This is the counter enzyme of PFK1 in process of gluconeogenesis. And for this fructose 1,6-bisphosphatase, never forget this fructose 2,6-bisphosphate is acting as a negative allosteric modifier. So you learn two things here. This fructose 2,6-bisphosphate is actually imposing the hormonal control on these pathways. This is going to increase the glycolysis by stimulating PFK1, and this is going to decrease the gluconeogenesis by inhibiting fructose 1,6-bisphosphatase enzyme of gluconeogenesis. Is it clear to you? So learn that. Very good. Now coming to next aspect, where I'll be talking to you about, okay. Right. You have got this table in your workbook. You see, this is a very, very important table which is talking to you about the difference between the hexokinase and the glucokinase. Right. So hexokinase and glucokinase, it's there in the workbook. Just see. These all points are very, very important for you, which is talking to you about the differences between hexokinase and glucokinase activity. As far as their functioning is concerned, both these two enzymes, whether it is hexokinase or glucokinase, it is converting glucose to glucose 6-phosphate, bachcho. Glucose to glucose 6-phosphate. And both of them, they are going to utilize ATP for donating the phosphate to glucose. But then they are isoenzymes, right? They are isoenzymes. When you talk about hexokinase, you talk about a number of isoenzymes, the type 1 isoenzyme, type 2 isoenzyme, type 3 isoenzyme, but glucokinase is the specific name given to type 4 isoenzyme of hexokinase that you must note. So glucokinase is not the different enzyme, it's not different from hexokinase. It's actually the isoenzyme of hexokinase only. That is the reason it is catalyzing same reaction. But when you talk about distribution, the glucokinase is found in liver and beta cell, and other places you find other isoenzymes of hexokinase. And glucokinase is being induced in presence of insulin, that you must know. And hexokinase is always there in the cell. It is not inducible. It is not inducible, right? It is not inducible. Now Km for glucose, very, very important point. What is Km? Km is the substrate needed to get the half max velocity. It depends upon the enzyme substrate affinity. If they have high affinity, less amount of substrate required. If enzyme substrate is having low affinity, more amount of substrate required. So this is a very important point which says that glucokinase is having low affinity to glucose, and hence it is having very high Km value, that is 180 milligram per deciliter of glucose. So liver glucokinase, pancreas glucokinase is going to be active and converting glucose to glucose 6-phosphate only when lot of glucose is available. But when you talk about hexokinase, always happy. Less than 1 milligram per deciliter of glucose is more than enough for getting the half max velocity when hexokinase is converting the glucose to glucose 6-phosphate. That is how you must know that. And the glucose 6-phosphate, which is the product of this reaction, inhibits which of the following? So it is hexokinase which is inhibited by product of this reaction, glucose 6-phosphate. Glucokinase is not affected at all, right? And I have told you the feeding and insulin is going to directly activate glucokinase activity. So please understand each and every word in this table is very, very important for you before going to the exam, one day before you revise this table, right? Okay, yes, Akriti, either you know or you don't know. So just don't worry, pay attention to the class, you'll learn so many things in biochemistry, right? Okay, and PFK1, this I already have told you. The PFK1 is under control of positive and negative allosteric factor. This is the same thing which is represented here. And I told you additionally, you write the reverse reaction, which I told you in the gluconeogenesis, you have the fructose 1,6-bisphosphatase activity enzyme. And whatever is positive here is going to act as a negative factor. And whatever is negative here is going to act as a positive control for fructose 1,6-bisphosphate. It's a contra-regulation, right? So contra-regulation. All right, very good. Now coming to next aspect. Okay. Before I come to PDH complex, I would like to tell you about two important effects. The Pasteur effect. What is Pasteur's effect? That's very strange effect. Strange effect in the sense everybody says that oxygen is good for health. But what you see here, that oxygen is having inhibitory impact on the glycolysis. This is called Pasteur's effect. Pasteur's effect, important one for you to know, that if a lot of oxygen is there, the glycolysis rate will be less. will be down regulated, right? So inhibitory effect of oxygen on the glycolysis is called Pasteur's effect. So do you understand it? Pasteur's effect, I just hope you write it and you can understand it as well. If you want me to explain you why this Pasteur's effect, how oxygen is having inhibitory control on glycolysis, for that I'll be telling you this. I'll take this space and okay. If there is lot of oxygen, there will be lot of ATP production, right? Or in oxidative phosphorylation, a lot of ATP production will be there and level of ADP and AMP will be lowered. So during presence of this excessive oxygen, a lot of oxidative phosphorylation is happening. That's going to create a lot of ATP. And what is ATP? What is ATP for PFK1? Anybody? I just told you it's a negative allosteric modifier. What is AMP for PFK1? It is positive allosteric modifier. So something which is negative factor is going to increase. So PFK1 activity will be down regulated. Something which is positive, that level is going to decrease in excessive oxidative phosphorylation. So the PFK1 activity will be down regulated. So guys, this is the explanation of Pasteur's effect, right? So just don't worry, pay attention to the class. You'll be getting all these annotated PDF, after this class. So just don't worry about it. Just pay attention to the class. So this was about the Pasteur's effect, which at times are being targeted. Now, this is more important, right? More important than Pasteur's effect for your MCQs. You must know Crabtree effect. What is a consequence of Crabtree effect? Anybody? Okay. Crabtree effect. The consequence is lactic acidosis, right? So what is Crabtree effect? See, the glycolytic pathway is a very, very beautiful pathway. If it is going at a fixed rate and with a proper supply of oxygen, everything will be good in the cell, right? But suppose if you're increasing the glucose in the glycolytic route without increasing the oxygen or increasing some of the intermediate of the glycolysis, like for example the fructose 6-phosphate, like for example the glucose 6-phosphate directly coming from some other pathway and getting dumped in the glycolytic route, but that time you're not increasing the oxygen, then it will result in relative anaerobiosis. Initial glucose will utilize the available oxygen, but because excess intermediates are jumping into the glycolytic pathway, the later intermediates which are entering the glycolytic route will not find oxygen and will be converted to lactate in anaerobic glycolysis. And this is a Crabtree effect. Why we talk Crabtree effect so much, and why I say it is very important for you to know, because number of diseases, when you talk about, listen carefully, number of diseases, like for example, when you talk about galactosemia, where galactose 1-phosphate is there, which ultimately is getting converted to glucose 6-phosphate. When you talk about fructose intolerance. So when you talk about von Kerke disease, where there is excessive glucose 6-phosphate for liver to utilize, all these conditions you have lactic acidosis, and directly or indirectly, Crabtree effect is the reason of lactic acidosis in these conditions. All right, very good. Yes, I told you, both are. See, increased in ATP is going to have inhibitory impact on PFK1, and decreased in AMP also will have down regulation of PFK1. So the glycolysis is going to be down regulated. Very good. Now coming to the next important aspect in the carbohydrate metabolism, which is favorite of your examiner nowadays. Every year they are asking you certain questions in PDH complex. So PDH complex is a mitochondria matrix complex where you have this particular complex, which is made up of 3 enzyme and 5 coenzyme. You must know their names. What is the 3 enzyme name and what is the 5 coenzyme name, right? And this PDH complex is doing what? PDH complex is converting pyruvate to acetyl-CoA So a lot of tamjham is required in the PDH complex. You need to have 3 enzyme, 5 coenzyme, which collectively is going to convert the pyruvate to acetyl CoE. And as you understand, pyruvate is 3 carbon and acetyl CoE is 2 carbon. There is a decarboxylation. So, and what is beautiful about this reaction is that even NADH is generated. NADH is generated. So this is called oxidative decarboxylation. This pathway is nothing but it's like oxidative decarboxylation. Oxidative decarboxylation, where NADH is also generated, carbon dioxide is also generated, and the pyruvate is converted to acetyl CoA, which is having next future in the TCA cycle. So coming back to this PDH complex, which is catalyzing this irreversible equation. Okay, थोड़ा हिंदी भी बोल दीजिए. I will say Shashank, हिंदी में भी I will tell you, but you just pay attention to the class and you will be coming across my Hindi as well. So PDH complex में 3 enzyme and 5 coenzymes, ठीक है? So 3 enzyme name is going to be pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. ये तो इंग्लिश में याद करना पड़ेगा, right? So these are the name of 3 enzymes which are required. And the one which is regulatory in this PDH complex is the first enzyme, the PDH enzyme. It's a regulatory enzyme. Other 2 enzymes are not regulatory, but regulatory enzyme of PDH complex is the very first enzyme, which is PDH, right? And when we talk about 5 coenzyme, we have got these 5 coenzymes which are needed in PDH complex, PDH complex. B1, B5 derived coenzyme E, B2 derived FAD, B3 derived NAD बच्चों, ठीक है? अरे क्या स्लो, सब स्लो ही चल रहा है. आपके पास वर्कबुक है, you have workbook in hand, you just add on these points if it is not there. And I'm sure in workbook your PDH complex is there. You see, this is the PDH complex. Mark here the name of 3 enzyme, 1, 2, 3, and name of 5 coenzyme, thiamine, lipoic acid. Coenzyme ASH, which is derived from vitamin B5, FAD, and finally NAD. So they will give you this picture. I'm sure they'll give you this picture, and they will, you know what they will do. They will delete some of the enzyme name and they'll ask you what is the name of E1, which of the following option is talking. Or they may ask you what reaction it is. You say oxidative decarboxylation. Or they may, English में तो पढ़ा रही हूँ. वो हिंदी मांग रहे हैं, मैं इंग्लिश में पढ़ा रही हूँ. I am teaching in English only, don't worry. So there are 5 coenzymes, right? So this I have told you, and the name of enzyme they will delete, and they'll ask you what is the name of this E1 enzyme, what is the name of this E3 enzyme, that you have to answer, right? And this cycle of reaction in PDH complex certainly tells you that if there is B1 deficiency, suppose B1 deficiency is there, the pyruvate will not be converted to acetyl CoA. So what will happen that time? If the pyruvate is not converted to acetyl CoA due to lack of B1, this pyruvate will be diverted to lactate. बहुत मजा आ रहा है बच्चों को. Everybody is enjoying so much. So pyruvate is converted to lactate. If pyruvate is not converted to acetyl CoA, it will say I will go and get converted to lactate. So the baby who is having B1 deficiency baby will have lactic acidosis, and that is explained only by this PDH complex requirement of vitamin B1. And Indian scenario, you see, a lot of poor families are there who cannot afford good vegetable, good fruits. So their child, they tend to develop the B1 deficiency, and those child present to the pediatric emergency unit with lactic acidosis, right? So just supplementation of B1 will help them. So this is a very, very important case which is presented to you nowadays. So pay attention to this aspect of it. Very good. Now coming to next. Okay, English बोलूं तो हिंदी, हिंदी बोलूं तो English. I mean, you people are confusing me, but I'm not getting confused. All right, इतनी English तो आती है यार. This much English you must know, right? Okay, so this was about the PDH complex, guys, and 3 enzyme name, 5 coenzyme name. I have told you the clinical aspect of it, that in B1 deficiency, the baby will have lactic acidosis. They will present a case of poor child who is coming with lactic acidosis. They'll ask you what supplementation to be given. You say B1 is to be given. B1 is to be given. Very good. Okay, what is the next page in your workbook? This is already there, my goodness. Everything I have given you. Now coming to gluconeogenesis. What important aspect of gluconeogenesis you need to know? Gluconeogenesis. In this picture, I have drawn the glycolysis on the left side and the gluconeogenesis on the right side. You see to it. See to it. So glycolysis, the same pathway which is there on page 1 is written here in the glycolysis. We have to pay attention to irreversible equations in the glycolysis because that only is to be shunted in gluconeogenesis. Remaining all reversible enzymes, mind it, listen it carefully, all reversible enzymes of glycolysis is used in the gluconeogenesis in the reverse direction. So if anybody asks you tomorrow the enolase, the mutase, the aldolase, whether it is used in gluconeogenesis from pyruvate or not, you say yes, it is used. Only 3 enzymes of glycolysis which are irreversible are not used in gluconeogenesis. Right, very good. So from pyruvate, when you're talking about the gluconeogenesis, what attention you're going to pay here? You see how the pyruvate kinase activity is shunted. Beautifully it is shown here. Pyruvate is converted to oxaloacetate, which then is converted to phosphoenol pyruvate. And you need to have 2 important enzymes here, the pyruvate carboxylase and pep carboxykinase. Pep carboxykinase is phosphoenol pyruvate carboxykinase, bachon. Right? If you want me to write the full name of this enzyme, I will write phosphoenol pyruvate carboxykinase enzyme. It's a favorite name for your examiner. They'll ask you what it is going to do. You say decarboxylation, right? Pep carboxykinase is not adding carbon dioxide. It's removing the carbon dioxide. Decarboxylation. It's a gluconeogenic enzyme. Pyruvate carboxylase is also the gluconeogenic enzyme, right? So these enzymes are important for shunting the pyruvate kinase reaction in gluconeogenesis. Then all the reversible equations of glycolysis used in the reverse direction. Just see the blue arrow here. Aldolase also used. And the counter enzyme for PFK1 is going to be fructose 1,6 bisphosphatase, right? And the counter enzyme for glucokinase or hexokinase is glucose 6-phosphatase, which is there in the endoplasmic reticulum, bachcho. It is there in the lumen of endoplasmic reticulum, may be a possible MCQ to you. Right, glucose 6-phosphatase enzyme in the lumen of endoplasmic reticulum, which is going to convert glucose 6-phosphate to free glucose by removing its phosphate from 6th position. Very good. So this is gluconeogenesis. And now I am coming to my PowerPoint slide where I'll be showing you the important aspect of gluconeogenesis, regulation of gluconeogenesis. So following 4 enzymes are regulatory in the process of gluconeogenesis. 4 enzymes. Right. What 4 enzyme? I told you pyruvate carboxylase, pep carboxykinase, fructose 1,6 bisphosphatase and glucose 6-phosphatase. All these slides you are going to get. The workbook annotated PDF also you are going to get. So just don't worry, just listen carefully. So these are 4 important enzymes which are going to regulate the gluconeogenesis. Right. And now if we are talking about these enzymes regulation, something very important comes up, and that is the role of insulin. Logically, you understand that insulin reduces gluconeogenesis, right? Insulin doesn't allow the gluconeogenesis to happen. This is doing so via its impact on the gene of pyruvate carboxylase enzyme. Pyruvate carboxylase is a protein, it's an enzyme, it's a protein which comes from a gene, and that gene is being repressed by insulin. So whenever insulin is there, it's going to repress the gene. Pyruvate carboxylase enzyme is not produced at all. When not produced at all, that's not going to lead to gluconeogenesis, right? So insulin is going to suppress the gluconeogenesis in this fashion, right? And on the other hand, you know logically, what glucagon is doing? What is glucagon? Glucagon is a hypoglycemic hormone. So during hypoglycemia, as many of you must be having hypoglycemia, your glucagon will be secreted from alpha cell pancreas, and that is going to increase the gluconeogenesis. So glucagon is going to induce the gene of pep carboxykinase, fructose 1,6-bisphosphatase, glucose 6-phosphatase. So this is a beautiful story of insulin and glucagon, regulating the process of gluconeogenesis. Regulating the process of gluconeogenesis, hormonal control of gluconeogenesis, I have told you. So if anybody asks you what insulin is going to do, how it is going to down-regulate the gluconeogenesis, you say it is going to repress the gene of pyruvate carboxylase. How glucagon is going to up-regulate the gluconeogenesis, going to induce the gene for pep carboxykinase. Remember 3 names. Pyruvate carboxylase is not coming here, right? So pep carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase. Very good. So this is a hormonal control of gluconeogenesis. Now coming to allosteric control of gluconeogenesis. See, all your MCQs will be targeted to these slides. These are key slides which you must listen carefully, remember, revise one day before exam, and you will do excellent in biochemistry. That's my promise. So now coming to allosteric control of gluconeogenesis, all these things you need to remember. Every time I ask you to remember, pyruvate carboxylase getting activated by acetyl-CoA. It's allosteric activator. So in mitochondria, if you have fatty acid beta oxidation wala, acetyl-CoA, it will activate the pyruvate carboxylase and upregulate the gluconeogenesis. Right? Very good. This you remember. Now coming to GTP. Which of the following is step in the gluconeogenesis? You think there is a requirement of GTP? Pep carboxykinase. It's a mitochondrial enzyme. You have just seen converting the oxaloacetate to phosphoenol pyruvate, pep carboxykinase, decarboxylating, needing GTP. Very good. Then, as I told you previously itself, whatever is positive regulator for PFK1 is going to act as a negative regulator for fructose 1,6-bisphosphatase. The AMP, the fructose 2,6-bisphosphate, which is going to be a positive for PFK1 is going to be negative for fructose 1,6-bisphosphatase. And ATP citrate, they are negative for PFK1, but they are activator for fructose 1,6-bisphosphatase. Who is going to remember this? सांस तो ले लो मैम, इमरान सांस तो ले ही रही हूँ. I am taking सांस, don't worry. Okay, I am slowing down. You note down these allosteric control for gluconeogenesis. Starvation fasting, I am coming to that. Don't worry. Write in your workbooks. Yes, Pratyush, you see how they are disturbing. Okay. Write down all these things in your workbook. What I've told you, I repeat it once again. So very, very important slide. You won't get everything together anywhere, right? So when we are talking about hormonal control of gluconeogenesis, we have got glucagon, which is required for gluconeogenesis. This is doing these things, going to induce the gene for these 3 enzymes. And insulin going to suppress the gene, and this is going to downregulate the gluconeogenesis. Coming to allosteric control, these are various allosteric controls. Acetyl-CoA, pyruvate carboxylase, GTP, acting as allosteric control for pep carboxykinase, and ATP citrate, and these story I have told you. Whatever is positive for PFK1 is negative for fructose 1,6-bisphosphatase. Insulin. Yes, insulin is going to inhibit fructose 1,6-bisphosphatase. It is very logical also to understand because fructose 1,6-bisphosphatase is the gluconeogenic enzyme. Insulin is going to increase fructose 2,6-bisphosphate level and thus going to decrease the activity of fructose 1,6-bisphosphatase. Very good. It's a crisp revision. I cannot afford to go very slow. Right. You already have workbook with you. So note down all these things there. Coming to next page in the workbook. What is that? Glycogen metabolism, where they're talking about synthesis of glycogen. What hormone is required? You tell me. Insulin is required, right? Insulin is needed for glycogenesis and contra-insulin hormone is needed for glycogenolysis. That is the first point you remember. Very good. And this is a picture which is talking to you about various steps of glycogenesis in a diagrammatic representation, diagrammatic fashion. What do you see here? You see the glycogen synthase enzyme, which is going to add glucose residues in which bond? What bond the glycogen synthase is going to make? Any one. It is alpha 1,4 glycosidic bond. Synthase is going to make you glycogen, partial glycogen, that is going to make you alpha 1,4 glycosidic bond. So this may be a possible MCQ. So it's going to make a linear chain. Synthase is going to make a linear chain, right? So what branching is going to do? Branching is going to create a branch by breaking the twig and fitting it in a branched manner, right? So branching enzyme is 1416 glucan transferase enzyme. This is very, very important for you. You must know branching is doing what? Branching is going to break 1,4 bond and it's going to connect it at 1,6 linkage. So this is going to create the branching. So tandem, somebody was asking me the tandem enzyme. Guys, this is a beautiful example of the tandem enzyme, right? So tandem enzyme is this. The meaning of tandem is what? Human walk is a tandem walk. One step after another step, then first step, then second step, right? So that's a tandem activity, tandem enzyme. So when you're talking about glycogenesis, you have got tandem enzyme, which is going to act one after another. And that tandem enzyme is glycogen synthase and the branching enzyme, right? Very good. Very good, very good. So it is a tandem enzyme. So this is needed in glycogenesis. So multiple times the glycogen synthase and branching will be acting in a tandem fashion to create a highly branched polymer of glucose, which is glycogen. Is it clear? Okay, so what is needed for glycogenesis? What is needed for glycogenesis? Glycogenesis require glucose in the form of UDP glucose, which is said to be nucleotide glucose. Naked glucose will not be effective. You need to convert the glucose to UDP glucose for glycogenesis. And insulin, as I've told you already, the hormone insulin is needed. What this insulin is going to do? Insulin is going to dephosphorylate glycogen synthase, which is the rate limiting enzyme, and this dephosphorylation of glycogen synthase is going to activate it. It will smile, it will act. So in presence of insulin, it is going to act. It's going to act. And rate limiting enzyme, I told you it's a glycogen synthase, and it's going to be active in dephosphorylated form, right? And branching enzyme is 1416 glucan transferase or transglucosidase. Coming to key points of glycogenolysis. For that, you just open the page which is talking to you about glycogenolysis. You can see this glycogenolytic steps. You have got these many steps. But what to learn in this glycogenolysis? Number 1 point. The immediate breakdown product of glycogen is not the glucose 6-phosphate. It is glucose 1-phosphate, which is converted to glucose 6-phosphate by phosphoglucomutase. And then this in endoplasmic reticulum is converted to free glucose. Simple. This is glycogenolysis. Only these things you need to remember. You need to remember the phosphorylase enzyme, which is the rate limiting enzyme of glycogen breakdown. You have got debranching activity. You have got glucose 6-phosphatase activity. All these enzymes are needed for breakdown of glycogen. So it is there in the workbook, and make certain notes in the workbook like these: glycogen phosphorylase acting as a rate limiting enzyme, and this is active in phosphorylated form. Again, the contra-regulation you see, the synthesis of glycogen needs dephosphorylation, breakdown of glycogen needs phosphorylation. Yes, vitamin B6 needed for glycogen breakdown, inorganic phosphate needed for glycogen breakdown, and this glycogen breakdown releases glucose 6-phosphate as 1-phosphate as immediate product. Many times they ask you, examiner think that you'll get confused and you'll say it is glucose 6-phosphate which is coming from glycogen. No, the phosphorylase action is going to release only glucose 1-phosphate. And phosphorylase, glycogen phosphorylase is also known as glycogen phosphorylase because it is cutting 1,4 glycosidic bond. It is called 1,4 glycosidase enzyme. PPT you will be getting, don't worry Sachin. Coming to, somebody was asking about fed and fasting type of thing. So this is a very, very important slide which is depicting you the source of glucose. Glucose as a fuel during fasting and starvation. Blood glucose we are talking. So when you take food and after that when you don't take food, then what happens? When you take food, that exogenous or dietary glucose is going to last for very little time. Dietary glucose is going to last for only 1.5 hours. Bachcho, after that is going to abolish from the circulation. Right? And when we talk about the glycogen as a source of blood glucose, we of course are talking about liver glycogenolysis, because muscle glycogen breakdown never gives you blood glucose due to lack of glucose 6 phosphatase enzyme in the sarcoplasmic reticulum. So liver glycogen is the sole source of blood glucose. And this breakdown, I'll be coming to the next slide. Its breakdown starts as early as 4 hours. It will be lasting for 18 to 24 hours. And gluconeogenesis, you can see this kind of peak is seen at around 2 to 3 days of fasting and starvation. After that, it's going to decline to a low level. So this is the picture which you're going to have, and this is the direct MCQ line. Dietary glucose lasting for 1.5 to 2 hours. Gluconeogenesis begins at 2 to 3 hours. Peak level of glucose coming by gluconeogenesis at 2 to 3 days, and it's going to decline at 7th day, and it's going to last for life. And when you talk about glycogenolysis, the breakdown starts around 2 to 3 hours, very early after fasting. You must have lunch at your lunch at 12 o'clock. So by this time your glycogen breakdown is already started, and that glycogen will be lasting only for 1 day at the most. Right? So at times they ask you that after overnight fast, what is the source of glucose in the blood? See, the dietary glucose, nil by this time. Overnight fast means 8 to 12 hours of fasting you're talking about. So after overnight fast, you do not have dietary glucose in the blood. And when you talk about gluconeogenesis, that will supply little amount of glucose, and the glycogen breakdown will actually provide you substantial amount of glucose in the blood. So that's a very favorite question of your examiner after overnight fast. Now overnight fast, when we say it, is 8 to 12 hours of fasting. So you have to say the first, if you have to choose only one answer, the glycogen as a source of blood glucose will be the best answer. All right, Namrata, this is not there in the workbook. These are additional points which you're going to jot down, right? Very good. Now this is about the source of glucose in the blood, right? Now coming to the next page, which is talking to you about galactose metabolism. Do you find this picture? Galactose metabolism, a very small, cutie and interesting pathway. Galactose metabolism, which is metabolizing galactose, and this pathway is seen only in the liver, you must know. Only in the liver, you must know. And this galactose is very easily converted to galactose 1-phosphate, then to glucose 1-phosphate, then to glucose 6-phosphate, which is further getting utilized. You need to have galactokinase enzyme number 1, uridyl transferase enzyme number 2, epimerase enzyme, that is 4-epimerase enzyme number 3. These are 3 important enzymes of galactose metabolism which you must know. And problem in galactokinase activity results in benign galactosemia, right? So benign galactosemia, which is not that benign because galactose is high in the blood, goes to the lens, aldose reductase sitting there to reduce the galactose, converting it to galactitol, which is going to absorb water and burst the cell, leading to cataract. So benign galactosemia also will have cataract as a feature, right? And when we are talking about uridyl transferase enzyme, its deficiency results in classical galactosemia, which is very frequently asked to you, characterized by repeated hypoglycemia and liver enlargement, hepatomegaly, liver enlargement, and of course the cataract. So this is a classical galactosemia. So anything may be asked to you. Yes, the cataract nature is oil drop cataract. I'll show you the picture. Can you see this cataract in galactosemic baby? This is an oil drop cataract, which is classically seen in galactosemia. Both in benign as well as classical galactosemia, you find this problem. And this picture of the baby is telling you about the classical galactosemia, where repeated hypoglycemia results in mental retardation. Cataract will be seen, organomegaly will be seen. Right. And these are the various dietary components which are going to provide you lactose and galactose. So big no to these compounds in a baby suffering with, these dietary compounds are not allowed in babies suffering with any form of galactosemia, right? So this is due to galactitol, which is produced from galactose. Enzyme name is reductase, aldose reductase. In diabetic patient, when there is a high glucose in the blood, the same enzyme is the culprit. Aldose reductase is the culprit for diabetic cataract, because that time the glucose is converted to sorbitol, which is again hygroscopic alcohol and results in diabetic cataract. But diabetic cataract is snowflake cataract. Snowflake cataract. Breast milk is also not allowed because that is also having lactose and galactose, right? So absolute no to galactose and lactose. Anyways, the galactose is dietary non-essential compound. Dietary non-essential compound. If mother catches you and says, Doctor Saab, if galactose is not given, then how the ganglioside will be produced? And ganglioside is so important for neurulema synthesis. So Dr. Saab, ganglioside is having galactose in it. So in galactosemic baby, when you are telling no galactose, then how the ganglioside will be produced? For that, the required amount of galactose who will provide? You say that required amount of galactose for making of ganglioside can be produced from glucose itself. In the nerve sheath cell, there is a very strong pathway which can convert the small amount of glucose to galactose in the nerve sheath cell, and that can be utilized for making of galactose. So big no to galactose and lactose in a baby with galactosemia. All right, very good. This is about the galactosemia. Now coming to certain MCQs. So who is going to answer me the MCQs? It is there in your workbook. Now let's start solving these MCQs. True statement regarding glycolysis and gluconeogenesis. What is true statement? RBC metabolize glucose by anaerobic glycolysis and in PP pathway is the correct statement. All other statements are wrong. Why? All the reactions in glycolysis are reversible is wrong. I have told you there are only 3 reactions which are irreversible, remaining all other are reversible. Fructose cannot be used for gluconeogenesis. It is wrong. Fructose is a carbohydrate. To call it as a gluconeogenesis, we need to have non-carbohydrate precursor. So fructose can never be a gluconeogenic substance. Glycolysis can proceed in absence of oxygen only if pyruvate is formed from lactate. This is again wrong. It can proceed in anaerobic condition only when lactate is produced from pyruvate. That is the correct statement, right? So this is how the answer is D in this. Now coming to question number 2. Which of the following statement about liver phosphorylase kinase is true? Liver phosphorylase kinase enzyme, which is responsible for phosphorylating phosphorylase enzyme. What is true? The answer is going to be it needs ATP for its action. I'll tell you how this enzyme acts. Right. Write down. This is glycogen phosphorylase enzyme. Yes or no? This is glycogen phosphorylase and this is also glycogen phosphorylase. But this time it is phosphorylated. Who has phosphorylated? Phosphorylase kinase enzyme, which itself require phosphorylated form. And with the expenditure of ATP, the ATP phosphate is given to glycogen phosphorylase, which is phosphorylating it, making it active. The phosphorylated form of glycogen phosphorylase is active. This is inactive. So you understand the role of this phosphorylase kinase, which is there in the question. It needs phosphorylation itself. It needs ATP for phosphorylating phosphorylase enzyme to make it active. Now let's see the question. We will see the question. Where is the question? Okay, this is the question. What is true? It catalyzes a reaction which needs ATP is a correct statement. All other are wrong here. It is present in the inactive form when epinephrine is elevated. See, when epinephrine is there, there will be phosphorylation of proteins, right? So that time it is going to activate the phosphorylase kinase. I told you just now. It phosphorylates to inactive form. Wrong, na? I told you. It phosphorylase to active form. It is phosphorylated in response to insulin. Wrong. It is phosphorylated in response to epinephrine. Right. Okay, very good. So answer for second question is C. Now coming to third question. A patient with von Gerke disease, that is type 1A glycogen storage disorder, can produce excess of ketone body due to all except. See, the fatty acid mobilization is actually high. Fatty acid mobilization from adipose cell is high. And that is only going for ketone body production. So what is wrong in this is option D. All other are correct because baby will have hypoglycemia. Lack of insulin is the trigger point for mobilization of fatty acid and ketogenesis. Oxaloacetate needed for gluconeogenesis. All are correct. All right. Very good. Very good. Now coming to fourth, you people don't pay attention to biochemistry. That is the problem, and you see it is scoring so important. So at least pay attention to this class, revise it 2 times. You'll do many questions correct in your exam. Infant with enlarged liver, glucose 6-phosphatase deficiency. This is again glucose 6-phosphatase deficiency means it is type 1A glycogen storage disorder. Cannot maintain blood glucose either by glycogenolysis and gluconeogenesis. This is absolutely correct because glucose 6-phosphatase is the enzyme needed for both the pathways. So this baby of von Gerke disease is going to have profound hypoglycemia due to lack of supply of glucose from liver glycogenolysis as well as gluconeogenesis, right? All other statements are wrong in this question. Where is all other statement? You see. Can use liver glycogen to maintain? No. Can use muscle glycogen? That is never possible. Can convert both alanine and glycerol to glucose? Not possible. I told you even gluconeogenesis is not taking place. Okay, very good. Cool. Coming to next question. Thiamine requirement increases in excessive intake of carbohydrates because I told you about the PDH complex story. That's going to convert the pyruvate to acetyl-CoA. Pyruvate comes from carbohydrates. So when you eat more of the carbo, you will have more of the pyruvate. You'll be requiring more of the B complex enzyme. B1 is being one of them. Okay, very good. At least you people are answering this so well. Very good. Coming to sixth question, fructokinase reaction. Fructokinase found in liver, first enzyme for fructose metabolism, converts fructose to fructose 1-phosphate. So what is the question? It is going to produce what? It's going to produce fructose 1. Never say it is going to produce 6-phosphate. That is by hexokinase action on fructokinase. So fructose by fructokinase converted to fructose 1-phosphate and fructose to fructose 6-phosphate by hexokinase enzyme. Remember this. It's not like glucokinase and hexokinase where every time you get to have glucose 6-phosphate. Over here this story is different and that is the reason they ask you this. Good, 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 good. Already marked. I only marked. Don't worry because it is important for me to know now and explain you quickly. Coming to lipids. In that the first fatty acid synthase complex picture is being given to you. This is fatty acid synthase complex. It's a dimer which is arranged in anti-parallel configuration. You can see cysteine overlapping with pentathene, pentathene overlapping with the cysteine. So anti-parallel configuration, head to tail configuration, and these two are fatty acid synthase dimers, and these are various catalytic activities which you need to remember. Unfortunately, you need to remember. Very good. Now, whenever we are talking about synthesis of fatty acid, it's a cyclical process. You need to have, for making of 16 carbon chain, for making of 16 carbon fatty acid, you need to have 7 cycle of fatty acids cyclical reaction, and each cyclical reaction consists of following 4 reactions. Following 4 reactions. One is condensation, then reduction, then dehydration, then reduction of double bond. Very good. So these are 4 reactions which are required for making of fatty acid: condensation, reduction, dehydration, and reduction of double bond. Now on the other hand, when we talk about beta oxidation, in this beta oxidation of fatty acid, which is predominantly taking place in the liver mitochondria, we have got 3 stages: number 1, number 2, and number 3 stage. First stage is the activation of fatty acid, which is nothing but converting the fatty acid to acyl-CoA. You can see in this. In this, first of all, the fatty acid is converted to acyl-CoA. This is called activation of fatty acid. After that, transportation of fatty acid across the membrane, mitochondria membrane, and the actual beta oxidation which is happening in the mitochondria matrix. So 3 stages you have got this beta oxidation of fatty acid. This is a very, very important structure which is talking to you. This picture is talking to you about carnitine shuttle. You need to remember this. Revise it one day before exam. Carnitine shuttle, outer membrane mitochondria, inner membrane mitochondria. 2 arrangement at outer acyl-CoA synthetase and CPT1. 2 arrangement at inner CPT2 and carnitine acyl-carnitine translocase, which is enzyme belonging to class 7 of enzyme classification. You can see in the cytosol itself, fatty acid is converted to acyl-CoA, which is next converted to acyl-carnitine in the space, which is going to go to the matrix only to reconvert itself to acyl-CoA. So revise this completely. This is a very important carnitine shuttle which is meant to carry or facilitate the transfer or transport of fatty acid across the mitochondrial membrane. Right? Very good. And one very, very repeated important point is CPT1 blocker. What is that blocker? CPT1 blocker is malonyl-CoA, which is produced in presence of insulin. During fatty acid synthesis, malonyl-CoA is the intermediate which is produced during synthesis of fatty acid. So whenever fatty acid synthesis is taking place in the cytosol, its automatic transmission across the membrane is inhibited. Why? Via inhibiting CPT1. Malonyl-CoA is inhibitor of CPT1, and this is the reason insulin is not allowing the fatty acid beta oxidation and ketogenesis to take place. Right? Okay, malonyl-CoA. Very good. So this is fatty acid transportation across the mitochondrial membrane by carnitine shuttle. Now when we talk about steps of beta oxidation, again you require 4 reactions. Steps in beta oxidation, you require 1, 2, 3, 4 reactions. Remember them. These 4 enzymes are needed again and again, and every time 2 carbon is removed from the fatty acid chain. So for completely oxidizing 16 carbon fatty acid chain, you need to have 7 cycle of such reaction. And acyl-CoA dehydrogenase produce FADH2 and acyl-CoA dehydrogenase, hydroxyacyl-CoA dehydrogenase produce you NADH. So every time, every cycle you are getting 1 FADH2, 1 NADH. So these things you must know. These 4 enzymes are important for each cycle of beta oxidation. How many cycle will be required for 16 carbon? 7 cycle will be required for 16 carbon chain beta oxidation. Is it clear or any doubt in this? Okay, so everybody, you ask me if you have any doubt in this. You need to know 4 reactions in fatty acid synthesis and on contrary 4 reactions in beta oxidation fatty acid. In fatty acid synthesis, 2 carbon is added in the elongating chain at the end of each cycle, and in beta oxidation, 2 carbon is reduced from the fatty acid chain at the end of each cycle. Very good. Now coming to lipoprotein metabolism. So in this HDL metabolism, LDL metabolism, VLDL metabolism, everything is being talked about. So this is the picture which is talking to you about VLDL and LDL metabolism. So what is the important core aspect of this picture, which is actually from Harper? This is the liver which is secretor of nascent VLDL, which is getting matured VLDL after accepting ApoC and ApoE from HDL. This is acted upon by lipoprotein lipase enzyme. You write it here, lipoprotein lipase enzyme is removing the fatty acid from triacylglycerol, and this VLDL is converted to remnant particle, which is either converted to LDL or it's cleared by the liver directly. And if it is converted to LDL, it is going to go to the extrahepatic tissue. So you must know these aspects of VLDL metabolism, right? Very good. Now coming to dyslipidemia, where the lipoprotein level is going to change in the blood. So you have these various dyslipopproteinemias, the type 1, type 2, type 2b, type 3, 4 and 5. And in this table, you mark the important defects which are responsible for these disorders. And most important for you is the type 2a and 2b, where the problem is in the LDL clearance. So LDL is not cleared either due to receptor mutation or for B100 mutation. The LDL is not getting cleared. So that's going to increase in the plasma, and that's going to increase the cholesterol level in the lipid profile. Very good, very good. Now, yes, coming to next point, dyslipidemia you learn properly. Coming to these questions, which of the following will be elevated at 4 hours after taking high fat meal? The answer is going to be VLDL. And for chylomicron, when it is going to appear in the plasma after taking lipid meal, it is going to be 2 hours after taking fatty meal. And VLDL, it's going to increase in the plasma by 4 hours. And VLDL has got nothing to do with the lipid diet. It is directly related with the carbohydrate intake. And chylomicron is directly related with the lipid intake. Right? Next question. Which type of lipase controlled by glucagon? Answer is going to be hormone sensitive lipase. This enzyme is getting activated by glucagon, epinephrine and norepinephrine. Epinephrine, norepinephrine and glucagon. Right? Very good. Now coming to third question. Hormone sensitive lipase mobilizes fatty acid. Inhibited by what? Inhibited by prostaglandin E1. So hormone sensitive lipase inhibited by insulin, prostaglandin E1 and nicotinic acid. Okay, nicotinic acid. Very good. So answer is D. Coming to fourth question. This is talking to you about total cholesterol value is given, HDL value is given, triglyceride value is given, LDL is to be calculated. So total cholesterol is the HDL cholesterol, LDL cholesterol and VLDL cholesterol. What is VLDL cholesterol? It's triacylglycerol by 5. So VLDL cholesterol is going to be 150 by 5, that is going to be 30 milligram per deciliter. So now LDL value is to be calculated. So LDL is going to be total cholesterol minus HDL cholesterol minus VLDL cholesterol. So that value is going to be, how much is total cholesterol? 300. This question you will be getting in the exam, don't worry. And VLDL is 30. So when you deduct 55 from 300, you get to have 225 milligram per deciliter. Maybe certain modification of the question. But this formula, which is called Fried-Wald formula, is very, very important, which you must know. Fried-Wald formula to calculate the LDL. Whenever triacylglycerol value is less than 400 milligram per deciliter, LDL is not being separately assessed. It is being calculated using this formula. But if triacylglycerol value is more than 400 milligram per deciliter in any person, that time this formula cannot be applied. That time LDL will be assessed by a direct assessment. All right, very good. Yes, VLDL cholesterol is divided, you can derive VLDL cholesterol by dividing the triacylglycerol value by 5. VLDL cholesterol, nobody is measuring. There is no kit to measure the VLDL cholesterol. And this same formula, even if TRAG is more than 400, you can use VLDL cholesterol calculation like this. Very good. Now coming to next question. After overnight fast, level of glucose transporter is going to reduce in. See, after overnight fast, insulin level will be high or low? Low. And insulin is needed for glucose uptake by GLUT4, which is present at adipose cell, heart cell, and skeletal muscle. And that is it. So 3 places you have GLUT4, which is insulin dependent. So these 3 organs will not be able to take the glucose whenever insulin level is low. So after overnight fast, the level of insulin is low, and adipose, heart and skeletal muscle will not be able to take the glucose from the blood. So answer over here is going to be adipocyte. Adipocyte. Very good, very good, very good. Oh yes, somebody is answering it so well. Okay, now coming to next question. In well-fed state, the carnitine palmitoyl acyl transferase on outer membrane, this already I have told you, is inhibited by malonyl-CoA. Yes. Now coming to important aspect of urea cycle. Very important, very, very important pathway which you must revise before going to exam. So urea cycle is this. Everybody knows that part of mitochondria and part of cytosol is involved. It's a multi-step process. And you can see the urea formation here. Read this pathway. We can't, Janvi, we cannot use the Friedewald formula if triacylglycerol value is more than 400 milligram per deciliter. If it is less than 400 milligram per deciliter, you can use Friedewald formula for calculating the LDL value. But if triacylglycerol value is more than 400, that time you have to do direct assessment. Yes? Okay, very good. So this is urea cycle, which I am not going in detail, but you are supposed to learn this whole pathway. And coming to triple H syndrome is seen in ornithine transporter defect. And one thing you must know, which is very frequently asked to you, OTC deficiency. OTC is ornithine transcarbamylase deficiency. This deficiency results in orotic aciduria. So if baby urine is having lot of orotic acid, direct hint is that it is a urea cycle disorder, OTC deficiency, right? Very good. Everything is important in the urea cycle. The first ammonia coming from glutamate, second ammonia coming from aspartate, and this whole cycle which is involving ornithine, citrulline, arginosuccinate, arginine, ornithine, you need to know this. You know, important, what is important, what is not important, stop asking this. Alright, because everything is important in your exam. Any question can come from anywhere. Alright, Siddhant. Coming to branch chain amino acid catabolism, that's a very good idea. You can do that. Now coming to branch chain amino acid catabolism. Right, so these are branch chain amino acid. This workbook is having this page with you. And this is given valine, isoleucine and leucine. Now I will just encircle the important points here. You have got branch chain keto acid dehydrogenase complex, deficiency of which results in maple syrup urine disease. You have got isovalyryl CoA dehydrogenase, deficiency of which results in isovalyryl coenzyme A is produced from the leucine, and deficiency of isovalyryl CoA dehydrogenase, this enzyme results in isovaleric acidemia. This is important for you. This is isovaleric acidemia. And you have got, what other thing is important? HMG-CoA. Can you see anywhere? HMG-CoA. This is HMG-CoA which is produced in the leucine catabolism. Right? So these are important points which you must know. Now coming to another pathway which is sulfur containing amino acid. This picture also you have. And pay attention to S-adenosyl methionine, SAM. Can you see SAM? SAM is the methyl donor which is produced in the methionine metabolism. And you can see homocysteine being one of the intermediate. So cystathione beta synthase, if it is deficient, you will have homocysteine urea type 1. Right? And excess homocysteine is converted back to methionine by homocysteine methyl transferase, the deficiency of which results in homocystinuria type 2. Good. Now coming to this picture which is talking to you about homocystinuria, where you can see the subluxation of lens. Lens is dislocated from its normal position, and that's very, very characteristic finding in homocystinuria. In tyrosine catabolism, this whole pathway is important for you because you have tyrosinemia type 1. Due to deficiency of this, you have tyrosinemia type 3. Sorry, type 2. And deficiency of hydrolase results in tyrosinemia type 1. And alkaptonuria, homogentisic acid oxidase, alkaptonuria, right? So alkaptonuria is due to deficiency of homogentisic acid oxidase enzyme. Yes, so these are the disorders which you must note. And when we talk about alkaptonuria, the urine turn black on standing. The fresh urine, this is the fresh urine which is colorless, but on exposure to air, it is turning black. And in addition, you have got deposition in the synovial fluid of these alkapton bodies, resulting in arthritis, black pigmentation in the conjunctiva, in the ear, the skin. These all are characteristic of alkaptonuria. Okay, now coming to the question, for metabolic disorder of urea cycle, which statement is not correct. So ammonia intoxication is most severe when metabolic block is prior to the reaction catalyzed by arginosuccinate synthase. Earlier the block, more chances of getting the frank ammonia in the blood. So this is the correct statement. This is the correct statement. We have to find the not correct statement, right? So we'll see what all correct statements are there. Clinical symptoms include mental retardation and avoidance of protein-rich food. This is also correct statement. Clinical sign is not acidosis, it is alkalosis. So this is the wrong statement. This is the answer. And aspartate is the source of second nitrogen in the urea cycle. All right. Now coming to the next question number 2. 2-year-old male child is brought with a pellagra-like feature and dermatitis. Elder sister is having the similar complaint. Pellagra-like feature, sister also having the similar complaint. It is seen in Hartnupp's disease. Hartnupp's disease where neutral amino acids are not absorbed. Neutral amino acids are not absorbed, right? So that will result in deficiency of NAD and pellagra-like features. Yes. Coming to third question, hyperammonemia blocking which stage in the Krebs cycle? Krebs cycle, alpha ketoglutarate is the intermediate which is going to capture the ammonia immediately. So that is going to block the alpha ketoglutarate dehydrogenase, which is the next enzyme. When substrate is like removed from the TCA cycle in cataplerotic reaction, of course there is no chance of having the action of alpha ketoglutarate dehydrogenase enzyme. Now catecholamines, which is produced by tyrosine, what is the answer? Production terminates with the dopamine in the brain but epinephrine in the adrenal glands. So very true, because whenever we are talking about catecholamines, it shows organ specific termination. So brain will have dopamine, adrenal cells will have epinephrine in the same pathway. And tyrosinase is not the initial enzyme, rather tyrosine hydroxylase is the initial enzyme for making of catecholamine. They are metabolized to both gluco and ketogenic. This is wrong. Tyrosine is getting metabolized to gluco and ketogenic. But whenever we are talking about catecholamines, it's converted to VMA, vanillyl mandelic acid, which is a waste compound, neither glucogenic or ketogenic. So this statement again is wrong. Contain methyl group donated by S-adenosyl methionine. All catecholamines do not contain methyl group by SAM. It is only epinephrine which contains methyl group, right? So all catecholamines do not contain. Okay, right, very good. Now coming to next question. Next question is fifth number question. Branch chain keto acid decarboxylation is defective in which of the following disorder? So you have branch chain alpha keto acid decarboxylase enzyme deficiency in maple syrup urine disease, right? So which is otherwise called branch chain ketonuria. Branched chain ketonuria, right? Branch chain ketonuria. See, everything cannot be taught no in 1 hour class. In first year, you are taking 1 year to learn these things, right? And in 1 hour, if you think that from scratch you'll be learning biochemistry, that's completely impossible. It is just a revision session. So those who have learned the biochemistry before this date will be certainly benefited. So just don't worry. Now coming to nicotinamide, nicotinamide is derived from tryptophan. Tryptophan is very, very important amino acid which is important for making of NAD. And during this pathway, you need to have vitamin B6, right? So B6 is needed for making of NAD from the tryptophan. You must know that also is very frequently asked to you. All right, so this I complete your revision of important metabolisms, and these pages you will be getting. Thank you very much, and I just hope you are benefited. Wish you all the best. Do it well. Read the question twice and then go to the option. Don't jump to the options directly after reading the question. You will forget whatever you know already, right? So all the best, lovely batch. And I just want all of you to get selected and come with flying color. Thank you so much.